Uh, good.
Hello, everybody. Rob Burgess here. I believe the webinar is now started, right? Right,
Yes,
Mohammed?
yes, that's
Okay,
right.
great. We are just waiting on the speaker. We had a bit of a technical hiccup here, and the speaker should be joining us in just a second. So I appreciate everybody's patience. There's our speaker now. We have Alfredo Quijano Rubio. We're going to let everybody get settled in to the webinar and just give it another minute or so. And as soon as everybody gets settled in, we'll go ahead and get started. Okay, we'll give it about another 30 seconds and then we'll get started. Okay, I think everybody's gotten settled in. So good morning, good afternoon, good evening, everybody, depending on where you may be logging in from around the world. I am Rob Burgess, and I head up business development for Sino-Biological. And it is my pleasure to welcome each and every one of you to the next installment in Sino-Biological's Lock and Key Immunodetection webinar series, where we hear about cutting-edge technologies as they apply to proteins and antibodies and their interactions and functions in nature, as well as in therapeutics and in diagnostics. We have an excellent speaker today, whom I'll introduce to you in just a minute. And the talk is focused on AI-powered protein and antibody/slash ELISA design. So I think you're really going to enjoy it. One housekeeping issue that we have is we ask everybody, number one, to type into the chat box, say hello, and tell us where you're from. We like to know where everybody is logging in from around the world. And then also, if you have questions for the speaker, type those into the chat box, and we'll withhold all those questions until the end of the seminar. And then I will walk through them verbally with our speaker and try to get to as many of them as I can. So with that, it's my pleasure and honor to introduce you all to our speaker. It's Dr. Alfredo Quijano Rubio. Dr. Rubio is a scientist and entrepreneur specializing in computational protein design. He is the co-founder and chief scientific officer of NovoBody, a company pioneering the design of novel diagnostics and research reagents using AI-powered computational design. He obtained his doctorate in bioengineering at the University of Washington under the mentorship of the esteemed professor Dr. David Baker, who was a Nobel Prize winner in chemistry in 2024. Dr. Rubio's research on designing conditionally active proteins for immunotherapy and biosensors has resulted in high-impact scientific publications, patents, and the creation of two companies, Neoleukin Therapeutics and, of course, NovoBody, of which Dr. Rubio is a founder. Alfredo has been recognized by the Business Insider and by Puget Sound Business Journal as one of the young leaders revolutionizing the health tech sector. And the title of Dr. Rubio's talk today is NovoBody and NovoLISA Platforms, Next Generation Antibodies and Homogeneous ELISAs by Computational Protein Design. Dr. Rubio, it's a pleasure to have you, and I will now turn the podium over to you.
Thank you very much, Rob. And thank you to all the attendees for coming today. I'm very excited to share with you today the what we've been building at Monad Bio. And hopefully we can find some applications to work on together. So please write down my email. Feel free to reach out after this meeting if you have any questions or you want to collaborate. With that, let me get started. So what I have for you today, I'll give you a brief introduction about our company, Monad Bio. And then I'll talk about the different technology platforms that we have developed in the past few years. This includes the NovoBody platform for de novo antibody mimetics, with a specific example of the anti-histag NovoBody, market leader in binding histags. Then I'll talk about the multi-specific NovoBody platform that we are pioneering now and some applications that are benefiting from this format. And finally, we are using our computational progress to make a NovoLISA, which is a de novo protein-based one-step immunoassay platform to help researchers and companies to do research faster and more efficiently. So a little bit about our company. So we focus on using AI-powered protein design to revolutionize diagnostics as well as research reagents. And we believe we're the only company really focusing on this market segment because we truly believe that good research, the foundation is the early research stages where we require exceptional reagents and exceptional tools. So we are bringing the revolution of protein design to this space. Our company was founded in early 2022. We're a spin-out from the University of Washington in Seattle. We have about 25 employees, and we have state-of-the-art computational as well as experimental facilities. Daniel Silva Manzano is our CEO and co-founder. Myself, Alfredo Quijano Rubio, I'm the CSO. We also have Greg Tibbetts on the team as our CFO, and Carl Walkie as our business development leader. And of course, we have Professor David Baker as one of our co-founders. And as Rob mentioned, he was awarded the Nobel Prize in 2024 for his pioneering work on protein design. As I said, our mission is to bring AI-powered protein design to build next-generation research tools and diagnostic assays. And my goal today is that you leave with lots of ideas on how you can implement this technology for your own use. And the idea behind this is that traditionally doing protein engineering has lots of limitations and it's a slow process. So it takes quite a bit of time to going from a natural protein that is suboptimal to an optimal version of it through iterative experimentation and evolution, because these natural proteins have lots of inherent limitations. However, with AI-based protein design, we can take a natural protein as a starting point and then use a computational platform to create a custom optimized version of this protein that has the right biophysical properties, the right function, and the right activity. And this can take as little as a few weeks to a month. The way we work as a company, we like doing protein design and we like partnering with companies that want to translate these products into real world products to market them, to use them, or to implement them in their own workflows. So we have more than 30 active global partnerships, out of which we have announced only 3 so far. And this includes key innovative in vitro diagnostics as well as research use reagents. As I said at the beginning, if you have any ideas or any applications, feel free to reach out because we would love to collaborate with you. So going a little bit deeper into our technology, we have developed multiple platforms. So I will summarize the different platforms right now, and then I will do a deeper dive on 2 of these later on. The first platform that we are going to highlight today is our NovoBody platform. Do not worry if you don't know the name of NovoBody. This is something that we have developed internally and coined. So hopefully in the future, you'll start hearing this concept much more often. So what NovoBodies are next generation single chain antibody alternatives that bind through 6 CDRs, which is a key differentiator compared to other technologies that you can find that use protein design. And we're applying this platform for research use and diagnostics as well. Then we have developed the protein mimetic platform for applications wherein you want to create a hyperstable protein, whether it's a cytokine, an enzyme, or an antigen, that you want to preserve a particular function, but you're having issues purifying it or because it's unstable. So we can apply this principle to improve the biophysical properties of the protein, but keeping the main function that wants to be preserved. We also have our LuxSit Pro reporter platform. So the LuxSit Pro is an AI-designed synthetic luciferase that we have commercialized. As far as we know, this is the first commercial enzyme that has been designed using AI and can be used for cell assays as well as other applications. And finally, one of the main topics of today is the NovoLISA homogeneous immunoassay platform which uses our luciferase reporter as well as either NovoBodies or antibodies to create very fast, simple to use, homogeneous mix and read assays that can be used for research use to facilitate your day-to-day research needs as well as for diagnostics. So going first, we'll cover the NovoBody binder part of the talk. I briefly introduced this, but basically NovoBodies, well, if we compare a traditional antibody to a NovoBody, on the left, you have the traditional antibody. These bind through 6 CDRs. That's how antibodies bind to their targets. They are about 150 kilodaltons, and they have 4 chains into a single molecule, which gives them very limited engineerability and are difficult to, in occasions, difficult to manufacture or optimize depending on the starting point. So our approach to this is we created the NovoBody platform, which also these molecules bind through 6 CDRs. However, they are only single chain, hyperstable. They're smaller. They are only 29 kilodaltons and can be very easily optimized and customized for your application of interest. And I will show you some examples about how we can do that optimization and customization. Importantly, the NovoBodies can also be expressed in E. coli, which brings a benefit compared to traditional antibody manufacturing platforms because it can accelerate the manufacturing, but also make it cheaper. So how does our platform work? So we start from an existing natural protein. It can be a very suboptimal antibody, SCV or VHH. And then we apply a list of requirements to our algorithm. This includes what is the desired isoelectric point? What is the desired amino acid composition? Do you need chemical handles for conjugation? What thermostability do you need? Any tags, any valency, and we can also tune the size. So with all these parameters, we use our proprietary algorithm, and in just a few hours to a day, we have a list of discrete leads that pass our computational metrics that we move on to testing. So we select a few leads per project, and then we express these directly into E. coli. So all these leads, in order to be passed, like accepted, they need to be expressed solubly and with high levels in E. coli. And then we do biolayer interferometry testing to make sure that these bind the target with the expected high affinity. From there, if the lead that we have identified needs to be further optimized, we apply a experimental optimization platform, which is really fast and really efficient at giving us significant boosts in expression if needed, but especially in affinity. When I was talking about optimization and customization, NovoBodies bring to the table a whole new dimension of customization that it's basically impossible with antibodies or even other proteins. So on the left, you're going to see different formats of NovoBodies that have been useful for different projects. So of course, you have a single NovoBody, which is 29 kilodaltons. Then we can start to combine them in a single chain to make a NovoBody duo to increase the avidity, as well as to make them bispecific or biparatopic to bind multiple epitopes on the same target, which significantly increases the affinity as well. Many people benefit from having FCs, so we can, of course, fuse them to an FCs. And as an FC fusion, these bring the benefit of adding more binding units compared to a traditional antibody, as well as having these in a single chain. So this antibody-like format only has one chain and can be easily expressed in any mammalian platform like HEC293s or CHO cells. And then we can start playing with added specificities and added formats because these express so well and are so stable that you get the benefit of creating the molecule that you need for your application specifically. Another layer of customization that we can provide that is especially useful for in vitro diagnostics is for site-specific functionalization. So at the bottom of the slide, you can see how we can design the NovoBodies with lysins on the surface, and we can specifically design for a number and position of lysins to strategically label the molecule with a given number of enzymes, probes, or whatever is needed. More importantly, we can actually remove all the lysines from the NovoBody if needed and only have one or multiple lysines far away from the binding site to enable efficient conjugation without losing any activity. So this is a very unique property of NovoBodies that it's just basically impossible with antibodies. And finally, we can do the same with cysteines. So our molecules do not have any cysteines by default. We design them without cysteines. So you can also put site-specific cysteines on the NovoBodies if needed. So let me show you some real-world examples of this. So we did a custom project for a major IVD company in the US where they came with actually very good antibodies. They had an antibody pair with high affinity, but that they require to modify to meet the needs of their assay development team and to change the format, basically. So they wanted to go from an antibody that had 88 lysines and 36 cysteines to a NovoBody formats that had the same affinity and specificity, but did not have any lysines on the binding region or any cysteines on the binding region, so that we could then add them site-specifically without interfering with the binding. The requirements were that they had to have a similar affinity to the parental antibody, of course, high expression in E. coli, which is also a differentiator compared to the original antibody. The isoelectric point needed to be lower than 7, melting temperature above 70, and then, as I said, the lysine-free properties, which is a very unique feature of our platform. What you see on the right are biolayer interferometry plots showing affinity. At the top, you have the antibody with a very good on rate and very slow off rate, which is desirable for high affinity molecules. And then the NovoBody in this format had very similar performance to the antibody, which is exactly what we wanted to see. But keep in mind that we completely changed the size, the format, the number of chains, the amino acid composition, so that this is basically a brand new protein. So the customer was really satisfied with this project. Moving on to a different application. In this case, the partner had issues with a fab because the fab after purification was showing some polydispersity, which you can see by secmols. You have a mix of monomers and dimers of this molecule, which is undesirable for a robust diagnostic development. It also had okay stability, but of course with room for improvement. And it actually had very good affinity for the target of interest. So the request was to make a NovoBody out of this fab as a single chain, only one cysteine for conjugation, and then it needed to be expressed in E. coli. And after putting it through our platform, you can see that it's 100% monodisperse, resistant to freeze-thaw cycles, higher melting temperature, meaning more stable, but it remains a very high affinity molecule. So all in all, a successful project. We are working with many companies and academic groups to develop partnerships. So far, we have announced 2 publicly. One of them with Biotechne, they licensed an AI design NovoBody Duo from us, and they are developing the product with it. And then the other one is a collaboration partnership with Gator Bio. They are the leaders in developing biolayer interferometry instrumentation to quantify protein-protein interactions, and we have developed a world-leading probe that I will show you in a minute. So one of our internal projects was we at the university were aware of the problems of anti-HISTAC reagents. I'm sure that everyone in the audience has expressed either proteins with HISTAC, has purchased them, and is familiar with the challenges in detecting them because the anti-HISTAC reagents are very low affinity and not very specific. So we set out to create a new anti-HISTAC reagent that would have much higher affinity and better performance than the existing alternatives. So we started from the very well-known antibody that has very, very low affinity. We're talking about 2 micromolar affinities. And then we created a NovoBody. The first iteration had very low affinity, as expected. And then we did multiple rounds of optimization until we go to our lead candidate, which has a picomolar affinity, which, as far as we know, is the highest affinity anti-histac reagent in the market as of today. The NovoBody has additional benefits compared to existing alternatives. Some of these include affinity, as I said. In this plot, in blue, you see the NovoBody performance, very good signal, very good on rate, very slow off rate, which is ideal for a BLI probe. In red, you have the second highest affinity reagent that exists. This is an antibody, but as you can see, there's a notable difference in affinity compared to our NovoBody. And finally, in green, you have the most cited, best sold antibody, anti-histac antibody in the market, which, as you can see, has very poor performance, which is, I think, very important, and the explanation behind why so many of your assays are likely not working with anti-histac reagents. Last but not least, the NovoBodies in blue show very, very good and constant stability, whereas antibodies tend to aggregate or unfold at different conditions, and then they never come back, which is a problem when storing reagents in your laboratory. So after developing this protein, we partnered with Gator Bio to create next generation probes for biolayer interferometry. And we released in collaboration the HisXT probes. These are available for your use if you're interested. And this shows you the benefit of using AI designed proteins bespoke for a given application. So since the proteins are smaller, we get higher signal on the BLI probe because the analyte has a larger mass compared to the NovoBody. Better baseline stability, and this is due to the higher affinity. And also it requires less sample because of the affinity and the size of the NovoBody. Faster loading, it's a result of the faster on rate. And then the stability provides more reliable regeneration. So these probes can be regenerated more times than the classic anti-histac probes. The feedback we're hearing from customers is that they're really impressed with the product. It really helped them move past some challenges they were facing when developing new assays or when discovering new antibodies. So this is just the first one of these products, but we're very excited to see what's coming, and we're open to more partnerships. If anyone in the audience is interested in trying the anti-histac NovoBody, please reach out. We have it on our website, but we're always happy to work on a new collaboration together. All right, so expanding the NovoBody platform, so we can make multi-specific reagents, and I want to show you a couple of examples of why this is so exciting. A little bit of basics on multi-specific antibodies. When we talk about bispecifics, this is a very lengthy and expensive process. So in order to make a bispecific antibody, you need to make you need to create 4 separate chains and then purify them through multiple steps until you get the right mixture of 2 different light chains and 2 different heavy chains, which is a lengthy process and results in an expensive molecule. That's why it's rare to see bispecific antibodies as research reagents as well as diagnostics. They are just too expensive. So we can bypass this problem by making NovoBody duos. So we can make a NovoBody and then combine them into a single chain to be multi-specifics. You just purify them as a single chain, whether you have an FC or not. It's the same. Simple manufacturing, you get much higher yields. And on top of that, you can continue increasing the specificity as well as the avidity, which gives you benefits compared to a traditional bispecific antibody. To validate that, we did this with already very high affinity NovoBodies for a given target. We had 2 NovoBodies that had an affinity of about 100 picomolar each. Then we combined them into a single chain, and we can observe an increase of about 10-fold in affinity. And then when fusing them to an FC and increasing the valency, we can see that this increases the affinity even further, which is a big improvement in when you're using a reagent. And this actually translates in real-world applications. So as everyone knows, detecting phospho-tau proteins, it's really critical in diagnosis of and research of dementia and Alzheimer's. So what we're showing you here is going from a monovalent NovoBody in blue increasing the valency gradually, and you can see in ELISA benefit in the limit of detection and sensitivity. So this is just to illustrate a very simple yet efficient way of improving your signal detection for a given assay. Another example that has been really useful for people to understand the power of the platform. So we also set out to create a bispecific NovoBody duo based on the therapeutic blinatumomab. For people familiar with this molecule, this is a notable protein because it's very difficult to manufacture, which results in a very expensive therapeutic. And the reason is because this bispecific scFv tends to aggregate and misfold when you purify directly from mammalian cells, and the average yield is really low. However, it works, right? So what we did without any optimization, we just put these two proteins through our pipeline, purify them. Again, no optimization. This is just side-by-side comparison. And you can already see a pretty significant increase in yield. But more importantly, this is mostly a monomeric protein. And after that, the protein has the same activity, affinity and cell-killing activity as the original blinatumomab. So again, just an illustration how the AI can help you improve your molecules that have limitations. So we are applying this platform to solve real-world, very important challenges, and we are working together with the Gates Foundation to develop proof of concept for two separate applications. The first one of them on the left, the Gates Foundation gave us a grant to develop NovoBodies for next-generation tuberculosis diagnostics, which has been a huge challenge to develop antibodies that are sensitive and can be applied in lateral flow for deployment of very inexpensive tuberculosis assays in low and middle income countries. So we are developing this. We don't have the data here today because we will present it later in the year, but it's looking very promising. As of today, we have developed NovoBodies that have much higher sensitivity than the gold standard in the tuberculosis space, which is really, really exciting. The other project is to develop a preeclampsia point-of-care diagnostic, which requires detecting two separate antigens and calculating the ratio, which requires, as of today, expensive and big instrumentation that can be only performed in central laboratories. So our goal is to use our NovoLISA technology to create an assay that can be read within minutes at the point of care. All right. So moving on to the second platform that I will cover today. So this is our LuxSit Pro and our NovoLISA technology. So briefly to introduce LuxSit Pro, so this is a de novo designed luciferase. This is in response to many customers out there that needed a small, stable, easy to manufacture reporter for luminescent assays. So the Baker Lab developed the first iteration of the molecule, and Monod optimized it and launched it commercially. So LuxSit Pro is the smallest luciferase and most stable luciferase out there that has very, very sensitive luminescent output. It is ATP independent, and it uses our own proprietary luciferase substrate, which is called luxtherazine. The molecule is cysteine and lysine free, which really, really helps when you want to use this molecule for applications for conjugation or applications in, for example, cell degradation, where you want to avoid any postulational modification on the reporter itself. And as far as we know, this is the first instance where a protein used for reporting has been designed with these features. Additionally, we have a split version that I will cover now that can be used to develop complementation-based assays. This kit is available, so please reach out if you want to implement this protein in your assays. So talking about the split assay, this is a reversible system made out of two protein components that we call alpha-lax and bit. The idea is that these two luciferase fragments do not come together and do not have activity unless they have induced complementation. And when they do, they actually give a massive signal increase up to 1,000-fold, which is very useful as a reporter. So these two fragments are very small in size, very low affinity, and can be easily fused or conjugated to your proteins of interest. So If you have any needs for implementing a luciferase assay to detect a given analyte, a given cell protein-protein interaction, please reach out because this is the perfect tool for that. And as I was saying, it's the perfect tool for that. And we have actually used it to develop our NovoLISA one-step homogeneous ELISA platform. So basically, when you think about ELISAs and homogeneous and immunoassays, sorry, you know that this takes a long time, right? So what if we could use AI to develop a better system that only takes a few minutes? So this is literally what we did at Monod. We created NovoLISA, which is the novel protein-linked instantaneous solution-based assay, which uses our luciferase as a reporter. So of course, we can use the NovoBodies as binders, but we have other options that I will describe later. But the conclusion here is that NovoLISA drastically compresses immunoassay analytical results. So it can give a quantitative, very robust result in just 30 minutes, as opposed to traditional ELISAs, which can take anywhere from 90 minutes all the way to 8 hours to set up, especially if you're making them custom and and they are not commercially available. So this is a huge advantage compared to running ELISAs, which in our experience can save a lot of time to academics and companies just running these assays constantly. Here's just one real world example of how this looks like. So we developed a NovoLISA to bind to detect CRP. So this assay is as simple as a two-step. So you add your biosensor mixture, which includes the two protein components, add them to a plate, add the substrate, and then you add the sample. And in less than 15 minutes, with a regular plate reader, this is the result you get. So this is time versus CRP concentration and different RLUs. So you see that the signal increases proportionally to the amount of analyte, is very robust, very stable. And you can read your analyte quantified through across multiple logs of concentrations, probably up to 5 or 6 logs, which is a huge advantage compared to ELISAs, which because of they use ODs, they are limited to 2 to 3 logs maximum. So if you have a need for a fast assay that can give you reliable data across different huge dynamic range, this is the tool that you want to use. However, many people wanted to use it and they told us, hey, but we have our own antibodies, we already have them in the lab. How can we make use of those and combine it with your luciferase to quickly make an assay that we can validate and test? So that's why we recently developed LuxLink. So LuxLink consists of ready to conjugate split luciferase with very simple chemistry and a very, very simple process. You can conjugate each part to each of your antibodies, and then you simply mix these two antibodies at the analyte, and then you can mix and read and get a immediate signal output out of that. The difference here is that now when you're making a custom ELISA that you need to create from scratch, that takes at least 6 to 18 hours with traditional methods. But now the conjugation to the LuxLink kit to your antibodies only takes 3 to 4 hours. Like with all the steps included that I will describe in a minute. But then once you have these pieces, it only takes 30 minutes to read. So if this is something that you're planning to do very regularly, you only need to do this part once, and then you only need 30 minutes to read once you have your antibodies, which would save a lot of time. The chemistry is very simple. The process any scientist can follow. Basically, we have the two luciferase parts, alpha lux, which requires a two-step process. We provide the ready to conjugate split luciferase, and then you just need to modify your antibody first, and then mix this modified antibody with the alpha lux piece and clean it up to get your purified alpha lux conjugate. So that's the more quote unquote more difficult part of the process. On the other side for the bit, we already provide this as an bioactive peptide, chemically active peptide, so you can easily conjugate it in one step, clean up, and you're done. So very simple process. Any scientist can follow it and do it successfully. This has been done for dozens of different antibody pairs in-house and by partners, and I can promise that it will work really well. We provide examples of gels on what to see at each step. And what you can see, it's at the very end of the process, you get very pure conjugate that can be used for your assay. The next step is doing a very small matrix of optimization to find the ideal concentrations of each of your components. I'm not going to get into the details, but just to show you that in just half a plate, you can do an optimization to find the best signal to noise ratio for your assay, which in this case, it would fall in these two concentrations. And once that's done, you can run a calibration curve and get results. I just wanted to highlight here that how simple it is. So you add the two antibodies, add your analyte for your calibration curve, shake and read. That's all it takes. It's a very simple process. So coming to the results, finally, after those two steps, then we can go to the results where here we're detecting IL-6 with the conjugated antibodies down to the 10 picogram per mil, which is a very sensitive assay for especially such a quick development process, right? Then on the right, what you see is that these conjugates perform really well across different matrices. So whether you're using this assay for serum samples or if you're using this assay for cell media, you can get very accurate, precise linear results across different logs of concentration as well. Finally, on the left here, we wanted to compare the performance when you have a conjugate versus a fusion, because this is a route that people prefer to take. It depends, right? So what we're showing you here is that both the fusions and the conjugates perform really well. So it's really up to you if you want to fuse or conjugate the luciferase parts to your antibody of interest. So coming up to the last couple slides here. If you want to try this kit, it's already available for purchase, but please reach out to me on our team. Here you have our emails. Whether you just want to try it out, please place an order. We can work together into giving this to you for evaluation. Once you get it, you will do optimization, and then you can decide if you want to commercialize this yourself. Then I will put you in touch with our BD team, or if you want to use it internally, we can also discuss different options. But our goal is that you can use these and that these create a better process for you. So we really want to make this available for everyone. So finally, I just want to give a brief summary of the different products or technologies that we have available, but also that we can collaborate with you for. So as a recap, we have the NovoBodies. We have the anti-histac NovoBodies available for purchase for different applications. They include different tags, different things that's really up to your use. But also, if you have any projects that could benefit from using NovoBodies instead of antibodies or optimizing your existing antibodies, please reach out. We are very happy to discuss. Then, of course, we have the luciferase, a very useful platform for your cell assays or in vitro assays. This is also available. And finally, we have the NovoLISA kits, either for conjugation or a bundle for conjugation and use, which we're very happy to discuss and give you the tips so that this definitely works in your hands. And with that, I want to thank the audience at the SinoBio team for the invitation and your attention. We really hope our goal is that these AI-enabled new tools get to your hands. So please reach out. We'll do everything we can to get this to you as soon as possible. Thank you so much.
Thank you, Alfredo. That was an excellent seminar. We really appreciate you taking the time to present it today. In the interest of time, we'll go ahead and get to the questions. I would like to remind the audience, if you have a good question for Alfredo, please type it into the chat box and we will walk through it verbally. I'll start with our first question from Sudeepa Bhattacharya. If I pronounced your name correctly, I hope I did. Sudeepa is asking about the expression of NovoBodies in E. coli and whether or not that expression affects their folding and functional performance, and how you might compare that expression to, for example, expressing them in mammalian cells, and would they be superior if you did express them in mammalian cells? Do you have any thoughts on that, Alfredo?
Yes, we definitely do. That's an excellent question. Thank you so much. So functionally, we do not see any difference or detriment by expressing them in E. coli, and that's one of the strengths of our platform, right? We are making sure that any leads that come through our platform can be expressed in E. coli without losing any affinity or performance metrics. And those, I think we showed two or three examples today showing exactly that, right? You take an antibody, you put it through our platform, express in E. coli, and then you get the same affinity and specificity.
Yeah.
That being said, some antibodies require PTMs, right, or glycosylations to function. Those are, I think, exceptions to the rule. In that case, if you need glycosylations, then you would need to go to mammalian cells. But to the second question, when we produce NovoBodies in mammalian versus expressed in E. coli, no difference. We routinely express NovoBodies in E. coli as well as CHO cells. No difference if we choose one platform or on the other, depending on the requirements.
Great. Thank you for that. I think practically too, that's wonderful because it's got to be so much cheaper and quicker to manufacture them in E. coli versus mammalian cells, right?
Yes, ultimately it depends on what you have set up in your facility, right? That's why we want to provide flexibility, and we've done extensive comparisons in both
Mhm.
systems because, yeah, depending on the company or the group, they have one system or the other usually.
Yeah. Yeah, it's a great system. The next question is from Dimitri Zorin. Please forgive me if I mispronounced your name as well. The diagram for NovoLISA showed two binders, one for each part of LuxSit. Does the system always require two binders to function?
That's an excellent question. So let me go back to the diagram. I guess we're referring to this one. Um, Thank you for the question. So It, I think this question depends on the exact target you're trying to detect. So here we rely on a sandwich format. So if what you need ultimately to do is bring the two luciferase parts into proximity, and for that you need two separate interactions to a molecule of interest. Now, many biological molecules have are multimeric, for example. In those cases, you only need one. And that's actually how we developed the CRP NovoLISA. So you can just bind the same epitope on a different molecule, and that will give you a readout. There are ways to circumvent that with other approaches, but for this to work, ideally you have either a monomeric um, target, and then you bind two epitopes, or you have a multimeric target, and then you only need one binder in that case.
Mhm. Great. Great. Thanks for that answer. Calling for more questions from the audience. Hisham, I know you're on the line and I know you might have some thoughts on LuxSit, so we'd love to hear your thoughts or your questions. While we're waiting for other questions, I just had a few. Alfredo, just one technical question. On LuxSit Pro, I noticed if I understood you correctly, you did site-specific recombinant exchange and changed out the cysteines and lysines so they couldn't be modified post-translationally in cells. Why did you pick just those amino acids? Because you know serines, threonines, tyrosines, many others get modified in cells. Is there a reason why you just deleted or replaced cysteines and lysines?
Yeah, that's a very good question. So the purpose of that, let me go back here. That's mostly thinking about conjugation, right? So
Mhm.
for conjugation, the most methods use uh, amine reactive moieties, like there are a few of those, right? And for those, lysines are the main target.
Oh, okay.
The other kind of option is modifying free cysteines, and that's why we chose those two residues.
Oh,
But
okay.
your question is more about postulational modifications, right? And that's a whole.
Yeah, yeah, like something that might affect its performance if it gets phosphorylated, acetylated, methylated, or something of that nature.
That's a very good question, and I think that's a separate topic, but we've also looked into it. So we've developed novel bodies for PTMs,
Mhm.
and when you develop an assay for that, you actually want to avoid your antibody to having those modifications as well, right? So that you can detect it
Mhm.
efficiently. So something very unique of our platform is that we can design the novel bodies without those motifs or those yeah, the motifs that will give you that PTM so that you don't
Mhm.
have any type of baseline noise just due to your antibodies. So that's a very good question, Rob, and a unique
Okay.
feature of our platform.
That's awesome. Another technical question. You probably mentioned this and I missed it, but in your AI input, the input of the information into the AI platform, can you just simply put in like a dissociation constant value, for example, and say, I need this level of binding for my NovoBody? Is that possible as well?
Um, it's a great question too. It's not possible at the moment. I think
Mhm.
the field as a whole, we are working towards that.
Mhm.
My take on the on that problem, and I think many PIs in the space would agree, is that first we require standardized high quality association and dissociation data so that we can train our models towards that. And I know many companies are working towards that future. But first, I think we need the data.
Mhm. Okay, sounds good. Don't blame me there. I think it's coming, and I think it will become that simple at some point, probably sooner than we think. Also, novel bodies clearly seem to be superior in many cases to monoclonals or recombinants. So I know you have the his tag offering, but are y'all planning on launching like a broader catalog of novel bodies that might replace some of the best-selling antibodies by companies like Sino, for example?
Well, that's a great question. So the way we see novel bodies is we really want to improve upon like improve the limitations of existing reagents. And I think the anti-histag was a clear example. And there are other products, there are amazing products that companies like Sino have, right? So for those, novel bodies might have more limited use. So we're really trying to focus on the ones that are very challenging or underperform
Mhm.
significantly the needs of users. To answer your question more directly, we've been focused on collaborating with companies. That's why we haven't released many more products. We're usually developing these in collaboration until these are ready, and then we co-commercialize them.
Mhm.
Um, but yes, so the answer is hopefully in the next few months, you'll see more releases of products. And
Mhm.
anyone that has any product in mind that they really need, please reach out because we would be happy to work on that as well.
Great, great. And we do have a question here from my good friend Hisham Zegzudi. Hisham is asking, is there any effect of serum or plasma on the split LuxSit light output?
Very good question. And short answer is yes, but it can be um, easily addressed. So I think I have one practical example here. So this is actual data. We decided to put very recent data to show you something that you might find in your laboratory. So when we perform this assay, what you're seeing here is comparing buffer signal, raw signal, right, compared to serum and media. You see a loss of, I can quantify this visually, but I leave it as a drop. However, with the buffer composition that we have developed, you can maintain the linearity so it doesn't affect your end result in any way. But it's a very good question. It's a known issue of luminescent probes, like the signal output is affected by the matrix.
Mhm. Great. And I think that's it from our audience. Just one last question. I saw the awards that were focused on human disease and disorders. Do you have a pipeline of therapeutics based upon the NovoBody technology? And if so, could you share any info on that?
Great question. We actually get that question all the time. So our core focus at the company is to specialize on research reagents and diagnostics because really no one else is, and there's a huge need in that space, right, for everyone. That being said, there's definitely an avenue to use novel bodies as novel therapeutics that could have a huge impact. We would welcome companies that want to partner to develop the therapeutics and work together on that.
Mhm. Sounds great. Wonderful business model there as well. So I think with that, we'll just wrap it up. First and foremost, I want to thank the attendees who joined us. We had a good turnout today from really from around the world, folks from India, from Greece, Brazil, and in the US, specifically Columbus, Missouri, and Madison, Wisconsin, and many other places. So thank y'all for showing up today. Alfredo, certainly want to thank you as our esteemed speaker. It's wonderful data that you've got there, and I just want to congratulate you and your team and everyone at Monode for all your success. And I know the future is very bright for you all.
Thank you very much. And thanks to everyone that attended.
Yeah, yeah. And also, I want to thank my colleagues, Ivana Ramirez and Mohammed Safarian, for flawlessly executing yet another Lock and Key webinar. We appreciate all your efforts. And with that, we will sign off and say good afternoon and good evening to everybody.